And I just wanted to show one more way in which to show this particular process. The one kind of area of this that we're missing would be the role of the ribosome involved in this. So we go from DNA, gene on the DNA is transcribed into an mRNA transcript. The mRNA transcript then leaves the nucleus, as you can see, and then begins interacting with the ribosome. As the mRNA transcript, this little blue ribbon kind of makes its way through the ribosome, it sort of excretes, in some ways, the polypeptide. And this, again, is just an overview. If you want a little closer view of this, it goes somewhat like this. You will have multiple ribosomes. You have a large and small subunit that comes along and they sort of meet together. And what's contained within that ribosome, you can see that the mRNA transcript is making its way not just through one, but several in a row. As long as you have an mRNA transcript, you can translate it into about 200 different proteins. So it's not just a one-for-one situation here. You can take an mRNA transcript and you can make multiple proteins from it just by running it through many different ribosomes in a row. And it's kind of interesting to show how this works. Actually, let me show you the kind of the inside of it. What the ribosome does is it brings together your tRNA, here it is, with its anticodon, which is already attached to the particular amino acid that will eventually be attached to the grouping here. Here's our mRNA transcript moving our way through. The tRNA will be attached within the ribosome. The mRNA transcript will run its way through the ribosome, and the tRNA will match up. You see the G for C, U for A for U, and will eventually put those amino acids and bond them together in this way. Here we see the first one, and then the second one, and then the third one. The tRNA releases after it has already dropped off its amino acid. Each tRNA will come into place and continue to bond the next amino acid in line, corresponding with the mRNA transcript codon with the corresponding anticodon of the tRNA. So in a practical sense, that's kind of a close-up view of it, but in a practical sense, what this means is that the ribosome will create will take the mRNA transcript, will create the growing polypeptide, which continues to get longer and longer and longer. You'll notice that the structure it's attached to, the organelle that's attached to, is the rough endoplasmic reticulum. And so what happens is this is being the polypeptide, generally speaking, will go directly into the RER, the rough endoplasmic reticulum. You might actually have some sugar groups, some carbohydrates attached right away. Remember that oftentimes you'll have a glycoprotein, which means a sugar attached to a protein, and then the protein begins to fold itself right there within the rough endoplasmic reticulum. And of course, you know that after this point, it's going to be pinching off into a transport vesicle where it will go to the Golgi apparatus, which will then produce further processing for the thing. But the main takeaway that I want from here is, first of all, of course, the central dogma of biology: gene transcribed into mRNA, mRNA translated into a polypeptide, an amino acid sequence that then folds itself into a protein. So that's one of the big takeaways here. And I think another big one is the idea that transcription is sort of the king of all of it. The transcription process, the transcription factors, allow you to have more mRNA or less. You can up-regulate a gene or down-regulate a gene. You can create more protein or you can create less protein depending on what that cell wants in that moment, what that cell is trying to do, what's important to the body in that moment, whether maybe it's important for this cell to produce more of whatever substance it produces, or it's more important in that moment for a cell to produce less of that substance.